Everyone for joining the Revance Fireside. We're thrilled to have Mark, Toby, and Jessica with us. Obviously, I'm joined by my colleagues, Stacy and Yorgi, the brains behind any operation, if you call that, at Cowen's pharmaceutical team here. So thanks, guys, for joining us today. A lot going on. Obviously, big transitions over the last period of time, last 12 months. And I know we have a huge potential opportunity in front of us in another major transition. So I thought the best way to start before we get into some of the groundwork that you've been laying with the dermal fillers and HintMD is to just take a step back, which we rarely do, and talk about DAXI and its differentiation, and maybe talk about some of the basics before we build up here on the dosing, the core active ingredients, really how DAXI is different than the other toxins, and clearly the leading toxin that's on the market, who will remain nameless for part of this conversation. So Mark, why don't I kick it off that way, and we go kind of some basic building blocks.
Great. No, I appreciate the opportunity, Ken, to kind of walk back, because I do think it is worthwhile when you start looking at, okay, why is it that we're seeing a differentiated duration profile? And if you go back, it was frankly by chance a little bit that the company came up with the product. So Revance started as a company trying to deliver their neurotoxin through the skin. And so in order to kind of get it to go through the skin, they needed to miniaturize and purify the toxin itself. So it went from a traditional 900 kilodalton molecule down to 150. And then they needed a carrier vehicle. So they found this peptide that was going to be the carrier to allow it to get through the skin. And so that combination of purifying it, reducing the size of the neuromodulator, and combining it with the peptide in order to be able to use it as a skin delivery mechanism was really kind of what drove to the product we have today. And if you think about the landscape, most of the other products, or all the other products that are on the market, have fairly similar performance profiles. And so it's a category that, while it's continued to grow in the market size, there hasn't been a whole lot of innovation around how do you engineer or modify the product to get different characteristics. And so we'd like to feel that we're going to be the first ones to come into the market in over 30 years and offer a truly differentiated performance profile driven by innovation and hundreds of millions of dollars. When you look at then that sort of transition from topical to injectable, what you've seen across a variety of different studies, both in aesthetic and in therapeutics, pretty consistent results. So if you look at our Sakura program, over 4,000 patients treated in the glabellar lines. If you look at our phase 2 upper facial line study, where it was forehead lines, crow's feet, and glabellar lines. And now you pair that with what we're seeing on our therapeutic side of it, most notably recently the cervical dystonia data and the phase 2 upper limb spasticity data. We're seeing a pretty good comprehensive group of data that suggests that it appears to be safe and effective, and that we're getting duration profiles out to kind of 24 weeks, depending on what the indication is. So we feel really good that as we, seek to get our first approval in the glabellar lines indication, that we're coming into the market with a pretty good body of data that supports the performance profile of the product.
Now, folks often talk, and I'm going to make a statement as I tend to do, and then ask a question. But a lot of the clinicians we talk to they say to us, no matter what the label looks like, they're not even going to look at it. And they're not being flippant. They're just saying, we really want to experience a product like this in our own hands, whether the label is incredibly positive, which we'll talk about what that means, versus, not having maybe everything that we would like. But from an investor standpoint, there is some focus around label. You just talk about the clinical studies that you ran. They seem to me to be incredibly similar to the other toxins, and very little, kind of differentiation in terms of how they ran them. Obviously, your data and longevity is better. But can you talk about why there would be anything lacking in the label, given those results that you have, in terms of why they wouldn't be included, and therefore give clinicians a sense of the duration that we've seen you experience?
Yeah, so I think that's a fair point, and clearly, a question that we've fielded from others. And, I'll take you back to kind of what we've been saying all along, is that we think that the clinical trials were well designed and were well run. And ultimately, it's that information from the clinical trials that finds its way into the label, and that ultimately provides sort of the foundation for what claims that you can make in the market. So clearly there's two different things. One is, as a company, what things can we comfortably claim, and that's going to be dependent on what's in the label. And then, as you pointed to, there's a market dynamic that can exist outside of it. But obviously, as a company, we're very focused on, getting data into the label that will allow us to promote the product based on a performance profile. If you look at sort of how we designed the study, there was, a 2-point efficacy threshold that we're looking to meet at sort of, the 1 month. And then we characterized duration over time as the loss of none or mild. There's been some debate and discussion around, what types of things of that data will find its way into the label. And what we've continued to stand behind is just, we would expect that the clinical trial information will be reflected in the data, and that the data that's ultimately in the package insert will form the foundation for what kind of claims that we can make. And so, we obviously don't have an approved product yet, so we can't say too much about it, but we continue to feel really good that, it was a well-run study, and that, the data that we generated, is consistent, as to your point, is what we've seen with other products out there.
Yeah. One of the other arguments, and I'm throwing them up and letting you bat them back down, but one of the other arguments is obviously that Botox, maybe you could increase the dose. A lot of folks talk about doubling the dose, and you started the conversation about explaining why Daxi is uniquely different and some of the formulation behind it. I always find that a little bit interesting, because we do know that there's dispersion
Sure.
with these products. So I'd want you to touch upon that. And then also there's a price on Botox. So doubling the dose would be already a price that's been established. And this is going to kind of then morph into the discussion around pricing of your product as well. I just don't think it would be that easy to charge double the price and give double the dose. And so maybe you can touch upon a couple of these issues, dispersion, and that argument that sometimes is made.
Yeah, well, maybe I'll hit the second one first. So to your point, I mean, is it economically viable? I mean, these products have been out there for, like I said, almost 30 years. And certainly physicians do experiment and have their different techniques that they use. And so if it was that simple and straightforward to get long duration by just upping the dose, we think we would have seen a lot more of that. And then there's a second question, is it economically viable if you have to sort of double the amount of product to get longer duration? There is some research out there that does say, more dosing can lead to longer duration, but it typically is accompanied by a higher side effect profile too, because you do get a little bit of, diffusion of the overall result. But, if you kind of then break it down further and say, all right, is there a way to compare these products apples to apples? And what we've gone back and as we looked at, everybody's dose is different. So there's no universal dose. We all make our own. And I use it as kind of an analogy. If I get a pint of beer in Ireland, the amount of alcohol there is going to be different perhaps than a pint of beer here in the States, right? So we all have our own unit of dose that we generate based on the LD50 assay work that we do. What you can look at is how much core neurotoxin